So what I want to talk about, these are kind of case studies from NeuroNEXT. So you've heard a little bit about NeuroNEXT if you joined our recent webinar about the various networks. And one of the things that is both one of the strengths and one of the biggest challenges in NeuroNEXT is we cover a broad range of diseases, as you probably heard about on the webinar. And we also focus on different types of studies, but mostly phase 2 studies, which is what I'm going to talk about today. Which, as a statistician, I've come to appreciate the more I've worked in NeuroNEXT, that phase 2 studies are some of the hardest to design in clinical trials. You know, phase 3 are complicated and hard to run, but if you've done what you need to do before you get to phase 3, the design of phase 3 should be pretty apparent to you. Phase 2 is kind of in this obtuse space where you're kind of wanting to show safety, you're kind of wanting to show like a hint of efficacy, but neither any one. One of the soapbox things I'll just say with that, that I'll allude to here, and if you want to hear me really rant, you can come to me later. You can never show in any clinical trial that the drug is safe. I know several people have said that. You can rule out different levels of safety, but drugs can be approved, and we still don't know if they're safe when they're given to millions and millions of people. So there's always this caveat when working with someone, if you want to show it's safe, how safe, right? And all you're going to be able to do is say it's not really, really bad, or it's not kind of bad, or it's not sort of bad, but you won't really know until it gets out there, right? So, one thing. So, a couple of things just to tie in. This was created in 2011 to conduct studies really across the neurological diseases, as I mentioned. Have to put this blurb on here, anybody can apply. One of the most common misconceptions, probably for NeuroNEXT and really the other networks, is that you don't have to be in the network to make a proposal to the network. So we try to say that as much as possible. We can do that. And we have like 3 types of studies. We also do biomarker studies. We're recently doing gene therapy studies, both in adults and in pediatric participants. But what I'm mostly going to talk about today is the phase 2 clinical trials, and in particular, this concept of go, no-go criteria, which was one of the key components when this was formed. You've heard, and several of you kind of even alluded to it, it's not just the field of neurology, it's the field of every clinical area, but in particular neurology, this has been a problem where there are things that look good in phase 1, look good in phase 2, and then don't hold up in phase 3. Some of that are things that couldn't be expected, but a lot of it could if you do kind of a post-mortem, where perhaps the earlier phase studies weren't as rigorous. This isn't new for me. Yeah. That's not somebody telling me to shut up. It isn't. That they weren't as rigorous, or in general, the thing that I think is the biggest concern is there's this concept in clinical trials called equipoise, which you've probably heard. But in essence, especially if you're doing randomization, it would be unethical to randomize participants to 1 of 2 treatments if there was not at least some level of uncertainty in terms of which is better or worse for you. Right? Equipoise is at kind of the broad societal level. I have yet to work with a single clinical investigator who was in equipoise for the project that they were designing and planning. Right? I mean, you would not be spending your time, your resources, your career on something that you didn't believe in and felt that it worked, right? But belief may not be reality. And so one of the challenges when you get to a phase 2 study or even some phase 1 study, or really a lot of the type studies that you guys are designing here, is if you don't have rigid criteria, when you see the data, you're going to see what you want to see in the data, right? You could show 2 people the same data. One who believes the treatment works will see positive benefit there. One who believes the treatment doesn't work will see nothing there. And so that's one of the problems where things can move forward with data that maybe haven't really proven anything or shown that there's a reasonable success, reasonable probability of success for phase 3. This is also, and this is something you could talk to Dietrich about maybe later, but one of the big differences in industry trials versus academic trials is in industry, usually it's part of a whole program. And every trial, every study has its place in the program. Academic trials don't have that as often as they should, right? Kind of the so what question, which maybe in your small groups you've been asked for that. What is the next step, right? If you're doing a trial and there is no next step, okay, it's great, it might be good for your career to learn stuff, but it's not really helping society on a larger level. If there is a next step, one of the things that needs to be part of your design of your clinical trial is what criteria do you need to meet to go to the next step? And more importantly, what criteria should be met that would prevent you from going to the next step? If there is no criteria that would prevent you from going to the next step, then it's really not going to tell you much about success or failure. And too many phase 2 trials that are initially designed fall into that latter category. They're basically set up that it doesn't matter if the treatment works or doesn't work, it's going to go forward. There's really no path to pull the plug on it, which then means you get to these larger, more expensive studies and the data doesn't look as promising as maybe you interpreted it in the phase 2 or earlier phase space. So I'm going to talk about just a few of the specific examples with studies we've had in NeuroNEXT and some of the go, no-go criteria that we've used. And you'll see less than half of our studies have met the go criteria. That's actually pretty good compared to whatever, if all of you in this room had your studies go to fruition and met like success criteria and went forward and it was proven in phase 3, one, we would be extremely happy. Two, we'd probably be really thinking this was an outlier of a class because the chances of that happening are pretty slim, right? I mean, the Knicks winning the NBA championship earlier was way more probabilistic than that, right? The Cincinnati Reds coming back and winning the World Series, way more probabilistic than that, Tracy, right? What? The train's scary. It's a freak. I like the Reds. They're not good. Okay, so going into this, the first one is kind of one that is not a high bar to set. It's a safety tolerability design, which are important, but also where you're trying to rule out various levels of safety. We had a study come to us in Huntington's disease in NeuroNEXT, the STARE study. This was looking at an investigational agent to treat irritable subjects in Huntington's disease. This is a side story, I have to tell this every time the study comes up, but this study had one of my favorite all-time animal models, mostly because what got me through my dissertation in my graduate school days were WWE and Jerry Springer, because I needed some mindless entertainment. But in the animal model for this, they basically took a rat, like a male rat, put a female rat in them, let them, get along and stuff. Then they took the male out, put another male in, and let the female get along with the male. And then they put the 2 males in and let them fight over the female, right? So it was basically an animal version of Jerry Springer, which is very entertaining. That's the only thing I remember from our investigators meeting, which is probably sad. But we actually did this to see whether in humans, right, where you couldn't put them on Jerry Springer and give them drug, although probably could have back in the 90s, looked at over a period of 12 weeks whether this drug was safe and tolerable. And in general, this showed that it was, right, which is kind of a very early study. This doesn't tell you anything about efficacy. This just says that it was safe and tolerable and is success to go on. But it does meet that go, no-go criteria. There was thresholds here that if it was sufficiently intolerable, it would tell you this shouldn't go forward, right? Another type of phase 2 study would be a surrogate endpoint design. Talk about biomarkers or surrogate endpoints. This is something where perhaps the phase 3 study is going to take a very expensive endpoint that takes a long time to obtain or some other challenge, and you have a good surrogate endpoint that can be collected in a shorter and cheaper time frame in order to assess the signal with the surrogate. If you see a signal with the surrogate, that might be your go criteria to look for the clinical endpoint in phase 3. I think this is a design that has a lot of appeal. It's a design that I think we'll see more in the future, but the rate limiting step here is we just don't have good surrogate endpoints for a lot of disease areas. And a good surrogate endpoint is not just a biomarker, it has to meet other criteria, and it can't just be correlated with the clinical outcome. There's a series of steps that typically have to be made to have a reasonable surrogate. We had a study like that early in NeuroNEXT in MS, the SPRINT MS study in 102, looking at an investigational agent, abudilast, which was an approved drug in Japan for asthma that was being looked at for multiple sclerosis, where the endpoint here was brain atrophy, so measured with an imaging endpoint. So even the phase 2 study was a big phase 2 study, but it was smaller than the phase 3 study would need to be. And in that study, we actually met the go criteria. You can see the curve on the left. It shows the measured brain atrophy over time, over a 2-year period, with those who received drug and did not. And you can see that with the red line, which is the drug line, the rate of atrophy was cut about half of what it was on placebo. More importantly, even though it was underpowered, on the right you can see the clinical endpoint, the effect over time. And there was a non-significant, but a also kind of meaningful trend there. You don't want to overinterpret because it wasn't powered for that, but having the statistically significant effect on the surrogate endpoint and the positive benefit seen on the clinical endpoint provided justification from this study to go forward. It has not for the company because there are other things that happen besides just one single phase 2 trial that determines whether a company moves forward with it. And I believe, my understanding, I don't know this, others may have a better sense of this, but they kind of looked in other indications and they kind of got away from NS with that treatment for various reasons. Another design which can be useful is a selection design. In its purest form, a selection design is just a race, right? You just put a bunch of doses or a bunch of drugs in a study and look at which one has the best effect and pick it. There's no testing involved with that. There are ways to make it more rigorous, which is kind of what we did in the study I'll talk about next, where you not just look at which one is best, but it has to be best or better than one of the groups by some fixed amount. So it has to be best and meet some threshold, for improvement. We looked at this in a study called Enlight PD or NN110 study, where this study was looking at 4 different doses of light therapy. 2 that were considered treatments, once weekly bright white light therapy and twice weekly bright white light therapy, versus 2 different controls, twice daily, it was twice daily, once daily, I think I said weekly. One of the controls was once weekly bright white, and the other was twice daily dim red light therapy. And the thought was so the go criteria here was that at least one of the interventions, the 2 interventions, had to be both controls by a certain amount. And then if so, you would look at the 2 together to see if twice daily was sufficiently better than once daily to justify the additional dosage, right? If it wasn't sufficiently better, you would do once daily because it's more apt to get people to comply to that. What we found was slightly interesting. You can see the results on the right. The study did not meet the no-go criteria. But this is one of those that illustrates an interesting thing that comes up sometimes. We talk about go, no-go criteria as if they're black and white, and they are in terms of are you ready to go to the next step, but not necessarily for the line of reasoning of whether the intervention is promising. This study did not meet its go criteria to move forward, right, with a dose and a control for a phase 3 study, which was what was intended. But it didn't, because what you can see there, the blue line is the once weekly bright white light therapy. And all 3 groups did better than that. So both interventions did better than that control. But the caveat in that study that is still being, I think, investigated is our other control, the twice daily dim red light therapy, also did better, right? And so you have the situation where it doesn't necessarily rule out this. It raises questions for why did the control not act like a control? Is it not just the type of light? Is it the frequency and the fact that you had this twice a day? Is there placebo effect because you're doing it twice a day? There's lots of theories that have been thrown around, but it's a situation where it's a different interpretation than if both controls had been lying on top of both treatment arms, which raises complication. But it clearly did not meet the go criteria as specified in the protocol. Another one, which I really like this design, investigators hate it, and it had a very poor PR in its early days of existence, which is now embedded, and it should be called something besides a futility design. I actually think a better non-negative name, and really what this is, it's a non-superiority design. What you're trying to do is show that an intervention is not sufficiently superior that you shouldn't take it to phase 3. What a futility design does is it basically looks at futility. If you think of when we compare 2 groups, typically what we want to see in a comparison of efficacy is we reject and say the treatment is better than control. And if you don't, there's kind of 2 other groups you could fall into. It could be worse than control, or it could be no better, or just a little bit better, but not enough to be statistically significantly different. What we're doing in a futility design is kind of flipping that around. And what we're looking at is it worse than treatment? And if it's not worse, it's because it's better, or it's maybe not better, or just a little bit better, but not enough to say that it's better. But what that can do, it becomes a gateway that if you can determine, is it not sufficiently worse, so that it's in one of those two groups in the middle or on the right side of that line, that would justify doing the phase 3 study to tease out, is it better, is it not sufficiently better? And so it is a gateway to avoid a word that some of you may have heard in your small groups, that if you use this word around any of us this week, you will see an instant reaction, which is underpowered phase 3 trial. Do not do an underpowered phase 3 trial. I can tell you, from being on study sessions, once the word underpowered phase 3 trial comes up, your study is dead on arrival. Dead on arrival, right? I've seen Tracy Glauser take knives out and stab proposals. It's so incensed. Be a charity that reviews. Don't have an underpowered phase 3. If utility design is a nice way to not have an underpowered phase 3 study. It doesn't say your treatment works, but it says there's at least a chance that it works. Okay. We looked at this with a study in myasthenia gravis, the BEAT-MG study, which looked at steroid-sparing effect of rituximab to see whether rituximab can get individuals off of higher doses of prednisone in the study, which would, in theory, help their clinical benefit as well, and would also justify doing a larger phase 3 study. You're flipping the null and the alternative here. So the null hypothesis is that rituximab improves the outcome by at least 30%, which is what ideally would be your clinically meaningful effect in a phase 3 clinical trial. And your alternative is that it doesn't. So what you're looking at here to reject the alternative and declare futility is that you are so unlikely to see your clinically meaningful effect that you can reject that in the phase 2 and say that it's not meaningful to go to phase 3. Whereas not rejecting the null in this study doesn't imply efficacy. It says there's a chance of efficacy that you could look further. A lot of when this study first came out, I believe Yuko was involved, right, in a study that looked at a lot of stroke studies that had failed in phase 3 and looked at if they had had a futility design in phase 2, they never would have made it to phase 3. So that's one of the big appeals, even though nobody wants to design their study to show futility. That's not really what you're trying to do here. You're trying to show it in a way that you can rule out futility and justify taking it forward to phase 3. But that word strikes up a lot of concerns. And this is kind of a sense of how it works, right? This is the power of the study. So this is the probability of declaring futility. If the if the drug was worse on the left side, that's the red. That's when you would want it to stop. If it's better, that's the green. That's just your type 1 error. And you have this area in the middle of yellow. I said that right. Yeah, by default, I say yellow, so I have to work really hard to say yellow. It's my southern heritage. And that's kind of what, if you don't reject futility, you're trying to determine, is it in the green or the yellow sense, right? That's really what you're addressing here. This study also met the no-go criteria, and actually, if you look, the blue and the red lines are in essence on top of each other. You can see here it had a P of 0.03. Our type 1 error was 0.1 here. Remember, in a futility design, you're rejecting and declaring futility. So what this study said was, based on this design, there was sufficient evidence that this study was not going to see a 30% effect if it was replicated in the phase 3 setting. That's a success, right? It's not a success for the investigator. It's not a success for the patients in terms of bringing treatments, but it's a success for the field of clinical research, and that you're not going to phase 3 and wasting resources to get the answer that the treatment would work after spending a lot of time and effort and resources in that space. All right. So the other groups of studies I'm going to talk about kind of fall into a different category, where you're either in phase 2 or you're using some type of novel endpoint or some type of novel combination of endpoints in some way to try to address the question. This was a study in cryptogenic central peripheral neuropathy, in N108, or the TOP-SPIN study, which was looking at topiramate, which is a drug that many are familiar with. It's been used in a lot of different diseases, but whether it could work in the cryptogenic sensory peripheral neuropathy, or C-SPAN population, in both two ways. With a clinical measure, which was the Norfolk Quality of Life Diabetic Neuropathy, QOL component, or intraepidermal nerve fiber density, so more of a nerve measurement. And this was set up with a placebo group and then an intervention group. And the GO criteria here It was, in essence, a non-inferiority study individually. And the go criteria was both endpoints. So the drug had to be non-inferior on both endpoints and superior on at least one of them. Didn't matter which one, right? So as long as both were non-inferior and one was superior to control, the study would meet its go criteria. In this study, it met the no-go criteria. And in fact, both of the endpoints were non-inferior to control, but neither proved superior to control, right? However, this is another one where the story gets a little interesting. If you look at the per protocol population, so per protocol population is different from the intent to treat. Per protocol is where you put in people who adhere to the protocol, stayed in the study, and so forth. If you look in that, it would meet the go criteria, right? Because you start to see a difference here on the nerve fiber density measurement. And what was interesting about this study in hindsight was a lot of, when we investigated what was different in the per protocol and the ITT population, it had to do a lot with a lot of early dropouts in the first 16 weeks, which is when dose was being titrated with topiramate, and due to, the AE was called, it was coded, it's measure-coded as cognitive disorder, which sounds worse than it was, but it was in essence lightheadedness, dizzy-headedness, things that you would expect with topiramate, right? But a lot of the topiramate group wasn't tolerating it early on and dropped out. Most of these were grade 1, most of them resolved really quickly. But because of that, so it kind of created this sense of, if you throw those people out, so the people who tolerated drug had benefit based on this. But this was not the a priori specified go criteria, so you can't necessarily take that to the bank. But in hindsight, in kind of the debriefing of this study, it was thought perhaps if you could do better up front of explaining the possible known side effects of topiramate, you might be dizzy, you might be lightheaded, these typically resolve over a few weeks to a month, and then perhaps there's long-term benefit. Maybe this would have turned out different. But it's another one of those where it did not meet the go criteria. But the no-go doesn't necessarily mean it didn't work. It means there's a nugget here, but you can't over-interpret it in one way or the other. But it leaves kind of the question open. More recently, and one of the studies, I think, with one of the coolest kind of designs and endpoints was our GNE myopathy study, and in 109, the MAGIN study that we just finished. This was a partnership with the Berry Consultants team, particularly Melanie Quintana, who had developed a disease progression model for GNE myopathy, which actually made this study doable and feasible. It reduced the sample size required to do a trial in this rare disorder by over half compared to the typical clinical endpoints. And so we were looking at a drug called Manac to see whether it slowed disease progression using this disease model. And you can kind of see here what the disease model does is it looks at the various muscles and kind of models them over time and puts it together. This one we just finished recently in November. The results were just presented at AAN for the first time. And it also met the no-go criteria, unfortunately, based on some promising results coming in and lots of hope. It didn't show anything. In fact, the probability, this gamma parameter is the parameter that showed if it's 1, if the treatment is the same as placebo, it's less than 1, it is better than placebo. And the probability was less than 1, and our primary analysis was 0.49 something, right at 50%, so basically a coin flip, which is saying that there was really not much going on there. So that one was a little disappointing. The other, which we have ongoing and is currently recruiting, is the Extinguish study. This is a study in NN111 or NMDAR encephalitis. It's been around for a while now, but if you've watched the Netflix movie or read the book Brain on Fire, this is that disease. So it's a very complicated disease. Again, it's a rare disorder. Here we're looking at a treatment with an immunotherapy, inebilizumab, or placebo that they're assigned to. You can kind of see the design of the study here. But this is also kind of a situation where there's some ideal evidence. It's a rare disease. We'll see how it shakes out. We're about 75% of the way through enrollment right now. Hopefully within the next year we'll complete enrollment, and then we have to follow the participants in order to close out the study. Both of these last two, the GNE myopathy and the NMDAR encephalitis, are examples of what I think are particularly challenging aspects of rare disease research, particularly in the academic setting, where really you're doing a phase 2 clinical trial, and they're funded as phase 2 clinical trials, right? But you also, in the back of your mind, have a sense, it's a rare disease where there are no treatment options. If the study is positive, there is at least a chance that it could become a registrational trial. But it will only become a registrational trial if it's positive, right? So you say phase 2 or phase 3 in disguise, right? So what that means is, you go to the Scooby-Doo thing, right? A phase 2 study, right? Well, let's see. I can't tell which one this is. So you get the result and it's negative. You take the mask off, really a phase 2 study, right? Or same concept, when you pull the mask off, it was a positive, and suddenly, I'm a registrational trial, right? That's a challenge when you're trying to organize this, because you don't have the resources to do a registrational trial and the resources to do a phase 2 trial are not the same. But you can't ignore that if you're doing a phase 2 trial that might become registrational, but you can't throw everything into it if you're doing a phase 2 trial that become registrational. So you're walking this fine line of knowing it could. As I refer to it in, again, my southern slang, it's a phase 2 till it ain't. Right? It may always be a phase 2. The NN-109 study fell into this category. It did not meet the GO criteria. It will forever be a phase 2 clinical trial, right? The NN-111 extinguish study is in the same way. If it doesn't meet its GO criteria, it will always be a phase 2 clinical trial. If it meets the GO criteria, the next step could be a phase 3 trial, but it could also become a registrational trial, which also means you have to be putting the procedures in place to prepare for that. So that's another reason why I say you have to kind of know where you're going when you're doing these early phase studies and what the next steps are or what the possible next steps might be. So with that, I think I will close. Again, glad to discuss anything about NeuroNEXT. Adam's here. Adam does the pediatric side of NeuroNEXT, but can also talk about any other aspects of NeuroNEXT from an NINDS perspective. Amy's here today as well, who is involved with NeuroNEXT as well. Myself, Dixie's involved with NeuroNEXT, Tracy's involved with NeuroNEXT. Dan's been involved with NeuroNEXT. I think he's still involved in NeuroNEXT. We don't let you go. Yeah, once you're in NeuroNEXT, you're in NeuroNEXT. And also, there's a QR code here if you want information about this. Right. So thanks again. Glad to discuss any more of this with you, and look forward to a productive week.